Hello, my name is Joseph Milliken, and today we're going to be talking about macromolecules in biochemistry, specifically carbohydrates and lipids. But first, we're going to start out with the 4 different types of macromolecules that you need to know: the monomers, the polymers, and the covalent bonds that make up these different macromolecules. So let's go ahead and get started. Okay. So I'm going to create a table. I have the type of the macromolecule. I have the polymer. Scratch that. I got the monomer. I have the polymer. And I have the covalent bond. I'm going to put C bond. It leaked down just a little bit, but I think I'm good now. Okay. So let's start with the first 2 that we're going to talk about today. First one being carbohydrates. Carbohydrates, what are they? They're sugars. Okay, so we're going to start out. If you know anything about sugars, one name for one type of sugar is called the saccharine. That's how I remember this. But for the monomer, or just one molecule of sugar, we call that a monosaccharide. Well, if it's a monomer, is a monosaccharide, what is the polymer? Well, shocker, it's going to be a polysaccharide. Makes your life just a little bit easier. Now, it's not like, sugar bonds, but it's pretty close for the covalent bond. The covalent bond is going to be called a glyco meaning sugar, lytic bond. Okay. Now, before I get ahead, remember that a covalent bond is where 2 atoms share electrons together, where a polar or ionic bond is one where one atom takes electrons and one atom gives away electrons. Okay, why is this important? Well, in organic chemistry, you're taught that ionic bonds are the strongest bonds that there is, with covalent being not as strong. However, in a biological environment, since these are aqueous solutions, meaning our cells and everything that's inside of us is basically an aqueous solution at all times, covalent bonds are going to be the strongest bonds that we're going to be dealing with. Okay, let's go over to the second one. And since we're going to be talking about lipids today, let's do it. Lipids. Okay, what are lipids? Well, lipids are fats. Okay, what are the monomers of lipids? Well, we have non-applicable. And you're like, what? Like, why are you teasing me with the non-applicable? Well, the thing about lipids is that they have tons of different functions and different structures, all sorts of different isomers, and since there's so many functions and structures of lipids, there's not really one that you could say like that's a mono whatever, or even a polylipid, or whatever you would call it. It's non-applicable, and the same goes for the covalent bond. Okay, so one less thing off your plate, you just got to remember it's non-applicable doing monomers, polymers, and covalent bonds for lipids. I'm sure you're asking, Joseph, don't forget proteins, right? Let's get into that, okay? Always in a rush, okay? Well, what are the monomers of proteins? Well, we call those amino acids. Okay. What about the polymers of proteins? Well, we call that polypeptides. Okay. Now, that might sound familiar. Why is that? Well, you might have heard of this thing called peptide bonds. Okay. These are the bonds that hold proteins together. which is a shocker because I was literally about to tell you that. The last one is nucleic acids. You've probably heard of the three above, but what about this one? What are nucleic acids? What make up DNA and RNA? Now, I would say the most important function, but if you ask Dr. Dini, who's our biology professor, he would tell you that that is not the case. He literally splayed out on a table in order to show us that, DNA is not the most important part. That proteins are, in fact, the most important part of the metabolism process, I guess, or mainly biological processes in general. What are the monomers of nucleic acids? Well, we call these nucleotides, which makes sense. Nucleotides. All of these are found in a nucleus, which is where DNA is, synthesized and replicated. What about the polymer? Well, we call these polynucleotides. Again, shocker with that one. And lastly, the bonds are called, get ready for this mouthful, phosphodiester bonds. And I'm not going to get into the organic chemistry behind phosphodiester bonds, but just know that they're the bonds of nucleic acids. It's what holds the DNA and RNA together. So I'm going to give you a second so you can look at the chart if you want. Pause it, do whatever you want. Okay. I always feel better second time around when I'm doing this because I did that the first time. And even then I didn't do great, but. So let's talk about carbohydrates. Let's talk about the structure. Let's talk about the function of carbohydrates. And first we're going to do it by talking about the two different types of glucose molecules, okay? You're like, glucose, it's sugar, okay? Carbohydrates, sugar. There's different types of sugars. One thing to know, if it ends with OSE, that is a sugar, okay? Like, we could have glucose, we could have fructose, we could have lactose. And all of these end with OSE, they're sugars. Okay? So I just mentioned that there's two different types of, ways to draw glucose. But when I say draw, they're two completely different things. They're not like resonance structures. Okay. First one is alpha glucose. Okay. Pretty simple. Alpha. And the way that we draw alpha glucose, actually I'm going to draw this a little bit lower so you can see better. Okay. The way that we draw this and the way that we draw both of the structures I'm going to show you are in ring format, okay? What do I mean by ring? Exactly this. So and remember that the formula for glucose is C6H12O6. And for all sugars, the formula is CH2O in parentheses with N being the subscript, meaning that you can multiply it by whatever integer, and that's what's going to be your formula. There's always going to be the same amount of carbohydrates and oxygens and twice the amount of hydrogens. Okay. But back to the structure. Okay. So I got a carbon linked to another carbon. Bring it up. Shocker, another carbon. So with only one oxygen being in this ring format. Now, what are attached to it? Well, to the different carbons, you're going to have alternating oxygen hydroxides, like on the inside of the outside of the ring. What do I mean by that? Well, I'm going to draw an oxygen in there, and I have a hydroxide, which what make up alcohols. Okay. I have a hydroxide in the middle, I have an oxygen. I have an oxygen in the middle, hydroxide on the outside. It gets a little bit different here. We're going to draw a carbon linked up to a hydroxide and then two different hydrogen molecules. Let me make sure I'm drawing that right. No, I told you wrong. This is a hydrogen. I should know better. But now I know. Okay. Nothing's going to be drawing that oxygen. Now, this is the important part. On this carbon on the far right side, for alpha glucose, it's going to have an oxygen on the top part and a hydroxide on the bottom part. Why is that important? Well, remember that we just said that the polymer version of carbohydrates are called they're called polysaccharides, right? Well, in order to make polysaccharides, you got to have multiple glucose molecules. Let's say that I have two glucose molecules bonded together. That would be called, I'll draw this in blue, that would be called disaccharide. Hopefully you can read that. Disaccharide, okay? Versus this is a monosaccharide, meaning one molecule of glucose. How is it going to bond? Well, first off, let's talk about the other structure. I'm not going to draw it all the way out. Instead, I'm just going to draw it out here like this. Okay, let's say that I have that carbon in the middle, okay, just like I have it right here. And instead of the oxygen being on top, the hydroxide is going to be on the top, and the oxygen is going to be on the bottom. This is what we call beta glucose, okay, just a B right there. Why is that important? Well, first off, when bonding these glucose molecules, alpha is not going to be able to bond to beta, and beta is not going to be able to bond to alpha. Why do you say that? Well, let's talk about how they bond. Let's say that I have right here an alpha glucose molecule on the other side. Okay, it's been flipped. I have a hydroxide and I have an oxygen bonded to a carbon. Okay. Well, when they bond, this hydrogen, the hydroxide right there, they're going to break off, with the product being, you guessed it, water. With two hydrogens, one oxygen to make water. What happens to this oxygen right there? Well, it's going to form a bond to this carbon. Okay, and this bond we call, it's called a strong covalent bond. That pen's going out. Get another pen. Go back to black. It's called a strong covalent bond, aka this is a glycosidic bond, just like we talked about earlier. Okay. Maybe I should write that out so that you don't forget. It's a glycosidic. Okay. And that's how these form up. This is how we create chains of saccharides or carbohydrates. Okay. Specifically, this is glucose. Why is that? Well, why wouldn't beta glucose work then? Well, let me show you. Let's say that I have, obviously we're going to flip it, so I have the hydroxide on top, and I get rid of the hydrogen right here. You're not going to be able to get water from this bond right here, and be able to form that strong covalent bond. So that's why it would not work. Okay, so an alpha bonding to an alpha. Okay, and there's obviously, this is alpha glucose. There's obviously like alpha fructose, alpha sucrose, so forth, and they form different structures. Specifically, for example, you might have a beta glucose and a beta galactose, and that will create what we call lactose, which is milk sugar. Okay, if you've heard of people being lactose intolerant. Well, they're intolerant to lactose, which is the sugar made up of beta glucose and beta galactose. Okay, remember OSC being sugar. Okay, let's talk about the process in which these bonds occur. There's a name for this. Okay, let's say that I break off this water right here, and I form that strong covalent bond. This whole process right here, I'm going to get rid of disaccharide. This whole process, it's called a polymerization reaction. Why is it a polymerization reaction? Well, remember that, if you remember from any of the lectures, or I'll refresh you, poly meaning many, Mary being the coming together, the forming of two different molecules. So this is one glucose forming with another glucose to form a disaccharide. Okay? And this whole thing is called a polymerization reaction. Okay? This is also called let me write this down, a dehydration synthesis reaction. I'm going to write RX in. Okay. Why is it dehydration? Well, we just took a water out of this, out of the glucose molecule in order to form that strong covalent bond, or that glycosidic bond, between the two different glucose molecules. Now, what if I want to break apart these glucose molecules? Well, think about it. We just took water out, okay? We dehydrated the glucose in order to bond them together. So you've heard of water-soluble molecules. Well, soluble meaning it breaks up in an aqueous solution or in water. So if I want to break up the saccharides and just have one monosaccharide, I would do the opposite. I would introduce water where that glycosidic bond is, and this would be called, make sure you can see it, hydrolysis. Let me make sure I'm telling you this right. Watch me tell you. I told him wrong. No, I said it right. Okay? Yeah, I told him right. Yeah. This would also be called a depolymerization. Yep. Reaction. Do you see it? Yes, you can. Sweet. I almost told you wrong, but I didn't. Hydrolysis is also called a depolymerization reaction. Depolymerization meaning the breakup of different molecules. Okay? And this is true of not just carbohydrates, but depolymerization, polymerization, that can apply to multiple different macromolecules. Okay. Now let's say I have a chain. Okay. Because eventually it's not going to just form a disaccharide. Okay. Hopefully you got that. Actually, I'll give you a second with it erased at the top a little bit. Okay. That way they can pause it and come back to it. Okay. Obviously, these carbohydrates are going to form chains, okay? You've seen it before with like that long chain, and that could be all different macromolecules. But like, let's say that this is a chain of glucose, okay? And let's say that I have around 10 glucose molecules. So I got like 1, 2, 3, 4, 5, 6, 7, 8. I'm gonna draw 9, 10. These are 10 glucose molecules in a chain. I know that they don't go diagonal, but you get it, okay? When we have around 10 or so glucose molecules and under 50, we call this an oligosaccharide. Okay? Oligo meaning few. So if oligosaccharide is like 10 or so glucose molecules, what is a polysaccharide? I know I've been throwing that term around a lot, but I'm gonna define it. Okay. This is 10 glucose molecules, right? Multiply that by 5 at minimum. So if I multiply that by 5, if I have 50 or more glucose molecules in a chain, that's called a polysaccharide. Okay? It can't be less than 50. It can be 50 or more glucose molecules in a chain. Okay? Not just in the environment, but in a chain. And I'll let you look at that real quick. Now you're probably thinking, and probably cursing me right now, Joseph, you just told me about alpha glucose and beta glucose. Why do I care? Well, alpha glucose and beta glucose have different functions. Okay, alpha glucose what are the functions of alpha glucose? Well, or actually, before I go into functions, what are different examples of alpha glucose? Well, first off, for alpha glucose, one example would be starch. Where do you find starch? Well, you find that mostly in plants. Okay. For example, have you ever heard of like a starchy potato? So a potato has a lot of starch in it. What makes a potato starchy? Well, its viscosity, I guess, or its texture is very thick. So, starch aids in the structure of plants, specifically plant energy storage. I might have told you wrong with the structure, but okay. Plant energy storage, and that's what we're going to see with alpha glucose. Alpha glucose is about energy storage. Okay, this is one of the functions of carbohydrates in general. Okay. What's another example of alpha glucose? Well, we have this thing called amylopectin. What is that? Well, we find it mostly in tree branches, plant branches, I guess. Okay. And lastly, and we just talked about 2 plant things, so you're like, Joseph, like, I could care less about plants, because I don't really care about plants either. What about in humans? Well, you might have heard of this. Glycogen. Okay. You can find this in the liver. Another place that you find it is in the muscle. Okay. Now, something to consider. With amylopectin, it's what we call branched. Okay. What do I mean by branched? Well, if I have these carbohydrate chains, they're going to branch out. Okay. With starch not really being, branched. Mostly it's just in large chains. But Glycogen is also going to be the same way. By the way, I misspelled glycogen. So, glycogen is also branched. Okay. Let's do it. Well, you look at it real quick. Now you're probably thinking, man, you told me about alpha glucose. It's fascinating. I can't wait for beta glucose. And get ready for this. Okay, so beta glucose with my fancy B. Okay. What are examples of beta glucose? Well, you might have heard of this, cellulose. And I told you wrong in the last part about starch. Cellulose is what aids in the structure of plants, specifically in the plant cell walls. Okay. Okay. Number 2, you have what we call chitin, not chitin. Our biology professor was very specific about not it being chitin. It's chitin. Well, what is chitin? Well, chitin, they're mostly in fungi cell walls. A specific example would be like a bug's outside shell. Okay, the more chitin that's in it, the more durable, the higher density, I guess, that the shell has for protection. Mostly in insects as well, so I'll put insects. The last one refers to bacteria, and we call this peptidoglycan. You might have heard of that too, at least it sounds familiar to me, specifically because it's made up of peptide bonds. Peptide bonds being, can you remember? Peptide bonds are for proteins. Right? Right. Okay. And we find this in bacterial cell walls. So quick overview. Alpha glucose is mainly for energy storage. Beta glucose meaning for structure. Okay. Plants, fungi, eukaryotic cells, insects, and bacterial cell walls. Okay, I'll give you a second to write that down. So even broader overview, the 2 functions of glucose are energy storage and structure. Okay, what about lipids? We just spent like a really long time talking about carbohydrates. You're like, boy, I'm dying to talk about that fat. Give me a second. Okay. There are a lot of different functions, a lot of different structures. Let's talk about 5 different lipids. Okay. First one, steroids. You've heard of steroids. Okay. An example of a steroid would be cholesterol. You may not have thought that cholesterol, or what lines up our arteries, and I know personally, my grandfather had triple heart bypass surgery to remove 99% of the cholesterol blockage in his arteries. But cholesterol is in fact a steroid. Okay. What about number 2? Phospholipids. What are phospholipids? You should know this. I hope you know it. Phospholipids make up the membrane of our cells, that phospholipid bilayer. Duh. Okay. Number 3, waxes. Come on, candles and so forth. It's a legit example. Beeswax, I guess. 4. I wouldn't have thought this either. But alcohols are also lipids. Now you're probably thinking, huh? We just saw hydroxides in the carbohydrates. Well, alcohols are lipids. So, just saying, there's lipids in carbohydrate chains. Just saying. Okay. Or at least hydroxides are a functional group for alcohol, so we're bound to have hydroxides and lipids. Okay. And we're gonna I'll show you that actually. Getting excited. Fifth one, prostaglandins. Okay. I'll let you look at that real quick. Actually, I'm gonna write on the side right here. As long as you can see it. What are the functions? There are a crap ton of functions, okay? It's actually 9 of them, according to DD. The first one being energy. Now you're probably saying, huh? Carbohydrates also do energy. I mean, look, when I'm getting ready for, a race, I might eat spaghetti the night before the race. That's mostly made of carbohydrates, not lipids. So what do you mean energy? Well, lipids do long-term storage of energy. And we'll talk about the reason why that's the case later on, okay, in this video. So stay tuned. Okay. Number 2, energy storage. Again, I will come back to that later in the video. Number 3, structure. Starting to see a little bit of a trend here. Shared purposes between the carbohydrates and lipids, but God has a reason for a lot of things, doesn't he? Number 4, signaling chemicals. What do I mean by that? Well, we just talked about steroids. Steroids also being, I'm going to mark this, hormones. And we mentioned prostaglandins. Prostaglandins are also signaling chemicals. Okay. Number 5, insulation. That makes sense, right? Let's face it, the fatter you are, the warmer you are. Okay, look at me. I'm super skinny. I'm always having to wear multiple layers. Okay. Pretty easy. There's 2 different types of insulation. Okay, thermal, just that layer, the density of fat being able to do that, but also electrical, which means that lipids are a good. They allow for a pretty good current to flow through them. Okay, can I mark down there? Yep. Number 6, padding. What do I mean by that? There's a lot of different parts in the human body that need padding. You want an example? This is horrible, but this is the one example that I remember, other than like behind your eyeballs, is there's padding behind this area, and you can imagine for what purpose. Okay, but that's made of lipids. Okay, so I'm not going to go specifically in it, but you get the picture. There's padding for all different purposes. Obviously, there's padding in your hand and stuff that's made of lipids. Number 7, and I'm thinking if I write over to the side here. Yep. Number 7, lubrication. Okay. Number 8, conditioning. You can think about it, your hair and so forth, but number 9, flotation. A lot of different functions of lipids, way more than carbohydrates, but all very important to organisms. Okay, so I'll give you a second to look at that. Kid you not, I just hit the wall. You know that feeling where you're just like, you're going strong and all of a sudden you're just like, man.
Well, you forgot a type of lipid.
Did we?
Triglycerides.
Oh, I'm going to talk about it. Literally,
Oh.
I'm just about to talk about
Okay. I was like, oh no.
it. No,
I mean, it's
that's
not like it.
because all it is 3 fatty acid tails.
Mm.
That's all it is.
Yeah.
So, let me give you an example of a lipid. Okay, we're going to draw it out, a function, and I'm going to tell you what type it is. Especially since it's important to know the nomenclature of these different types. It's pretty simple. Okay, these are made of like a bunch of carbons linked to each other. See that I got 3 carbons right there with hydrogens being attached to all of them. But what I'm going to attach below, just like we talked about hydroxides being used for alcohols, I have 3 carbons with 3 different hydroxides linked to the bottom of this. And what am I going to do after that? Well, I'm going to do like this little squigglies. You're like, what the heck is that? Well, that, my friend, is a fatty acid tail. Okay. What are made of? Okay. This is where I cut it. What do you call this? Well, we call this a triglyceride. Remember that we said the monomers of lipids are non-applicable and the polymers are non-applicable. The bonds are non-applicable. So I want to add a little bit of simplicity to your life. Tri, meaning the 3 different carbons right here. Okay. What if I had 2 carbons? Well, I'd call it diglyceride. What if I had 1? It would be monoglyceride, I guess, or just glyceride. Okay. But this is triglyceride. What is this used for? Well, or actually, what is it? Solid fats are all made of triglycerides. Okay. So solid fats. What I mean by that? At room temperature, they're solids. Okay. This is where I cut it. I gotta look at my notes real quick. This is actually, I'm expanding a little bit because he talked a lot more in detail. Man, I wish I'd gone through this. A lot more stuff in the video.
My friend just went to the melting pot.
What is that? Is that a bar?
No, it's a restaurant. It's like a fondue restaurant. And his balance was $123.
For melted cheese?
Maybe chocolate. I don't know. Maybe they have chocolate fondue.
It's an expensive chocolate, man.
Have you ever been to Crave?
No.
It's a place here, and they do like a little fondue thing too. It's not that expensive.
Not
Or they do like
120
in-house, like make your s'mores. Like they'll give you like a platter and you make your own s'mores.
Yeah. So now we talked about in carbohydrates, the dehydration / hydrolysis reaction. So the same thing is going to happen with lipids. Oh, obviously, when I say that, it's a little bit different. But let's say that I have another hydroxide. Let's see if I can get away with a dark blue. Can you see that? Yes, you can. Okay. say I got this hydroxide right here. Well, just like we had in the last reaction, or in the carbohydrates, we could take water from this, okay? That being a dehydration reaction, okay? What is that gonna form? Well, I got a chain, a carbon right there. This is actually what makes up these chains right here, hydrogens being on the sides. Now you probably heard of saturated and unsaturated fats. What does that mean? Well, the more hydroxides that you have, like let's say I had that right here, the more hydroxides you have on the sides are connected to these carbons, the more saturated it's going to be. Why is that? Well, think about it this way. We just use hydroxide to make water. Okay, combined it with another hydrogen off of our backbone. Let's use the right terms. The backbone of our lipid. So that's what makes it saturated. Okay. And so when we talk about saturation, we saturate with hydrogen. Okay. Let me draw it real quick. So now we're going to get, we're straying away from structure. We're going to talk about some, I guess, theoretical stuff, mostly just general information. Remember that I said that one type of lipid is called a prostaglandin. And if you're like me, who's never heard of a prostaglandin, you're probably thinking, well, what is that for? Well, there's three different deals. It's a local hormone. Okay. What is it? Well, it's used for smooth muscle contraction. What is it also used for? Well, it's also used for the synthesis of pain receptors. I would say that prostaglandin is pretty important, specifically for this last function. Why? Well, pain is a very important part of our lives. It tells us, when something's hurting. Obviously, chronic pain is not good, but without prostaglandins, we wouldn't be able to tell if something is wrong. We wouldn't be able to treat it. So, obviously, very important. And finally, I mentioned earlier that we would talk about the difference between carbohydrates and lipids. So, in terms of energy, what makes a carbohydrate different than a lipid? I'm going to let you look real quick. I'm going to erase this because there was so much stuff to talk about. Well, first off, when you look at for molecules, okay, or for grams, okay, for 1 gram of, I'm going to put C, but carbohydrates, there is actually equal sign 4 calories. For 1 gram of lipid, there's 9 calories. Twice as much calories for 1 gram of carbon. So there's twice as much, more than twice, the energy. Which means that lipids are not inefficient. They're a more efficient energy producing molecule. Okay, not producing, but carrying. How about that? Okay? Thus, they're great for storage. those carbohydrates are going to get burned off pretty easy. So if you want energy for later on, that lipid is going to be very helpful. Okay. This is why animals have fat, why they carry fat. When they're chasing around their prey, they need to have as efficient energy as possible. And in terms of, I mean, let's face it, why do you think cross-country runners wear skimpy clothes? They're trying to cut weight. Same thing with animals and fat. They're using the most efficient energy-carrying macromolecule in order to maximize their ability to go hunt their prey. And that's why, that's what the difference is between lipids and carbohydrates. I hope that helped you. The next video we're going to talk about amino acids and their polymerization.